Welcome to This Explainer. I am absolutely thrilled you're joining me today because, well, we are looking at a truly groundbreaking discovery. We're going to unpack a brand new study that is literally rewriting our entire understanding of cancer immunology. For decades now, we've relied on certain assumptions about exactly how the immune system fights tumors. But recent data, it's flipping the script entirely. So, get ready for a biological detective story that fundamentally changes how we view the human body's ultimate battlefield. Okay, let's just dive right into this by posing a really fundamental question, one that has stumped researchers for years. How do immune cells actually know what to do once they're inside a tumor? Do they arrive as these blank slates, just taking orders directly from the cancer? Or do they show up to the fight already knowing their mission? To figure this out, we really have to solve the core mystery of how these cellular soldiers get their orders in the first place. So let's start with Chapter 1, the immune cell mystery. Think of this as basically an unsolved biological cold case. For a long time, we've been missing this absolutely crucial link regarding how these cells travel through the body. And to understand why, we need to quickly look at the two main branches of our immune system. On one side, we've got adaptive immunity, your T cells and B cells. These are relatively easy for scientists to track because as they develop, they undergo genetic reshuffling. They basically print their own unique genetic barcodes, which allows us to trace exactly where they came from. But then on the other side, we have innate immunity. This includes myeloid cells like macrophages. These are our first responders and they are highly, highly abundant in tumors. The problem? They don't have those built-in barcodes. They are completely anonymous. Tracking where they come from and what their family tree looks like has been historically impossible. Well, until now. Which brings us to Chapter 2, MitoTrek, the biological barcode. To actually crack this cold case, researchers at Stanford and Memorial Sloan Kettering had to invent a totally branding forensic method. They call this revolutionary tool MitoTrek. You can kind of think of it like an ancestry test, but for a single cell. Because mitochondrial DNA mutates at a pretty high rate, it acts like a natural ticking clock. By mapping out these specific mitochondrial mutations, scientists can finally reveal the family history and the clonal lineage of these once anonymous innate immune cells. And the elegance of MitoTrek is really in its process. It's essentially 3 steps. First, they isolate single cells directly from human tissue. Second, they read their chromatin accessibility, meaning they look at which parts of the cell's DNA are open and active, so they're essentially reading the cell's current job description. And third, they simultaneously sequence the mitochondrial DNA to find those unique, naturally occurring mutations. By combining the cell's current identity with its mitochondrial ancestry, they can confidently group these cells into specific family clones. And this brilliantly illustrates the sheer scale of what they accomplished here. 218,715. That is the number of individual, single cells profiled in this study. To ensure absolute forensic accuracy, the researchers didn't just look at a handful of cells. No, they processed hundreds of thousands of them across multiple patients. This gave them an incredibly robust, high-confidence map of the immune system's family tree. Let's move on to Chapter 3, Mapping the Tumor Battlefield. Now, we transition out of the lab and into the very real, intensely complex environment of the human body. The researchers applied MitoTrek to tissue samples from real patients who were actively fighting early-stage non-small cell lung cancer and ovarian cancer. And importantly, they didn't just look at the tumors themselves. They also sampled healthy tissue right next to the tumors to compare how immune cells behaved in a war zone versus, say, a peaceful neighborhood. But they didn't stop at the local tissue either. They also took samples of the patient's peripheral blood. And this introduces a really vital concept for us: the battlefield versus the supply line. If the tumor is the battlefield where the heavy fighting happens, the blood flowing through this vial is the crucial systemic supply line, delivering the troops. The big question was, how are the troops in the supply line actually related to the ones fighting and dying on the battlefield? Well, when they mapped the family trees across the blood, the healthy tissue, and the tumor, the results were honestly staggering. Looking at the clones shared across multiple tissue sites, a massive 27% of myeloid clones were found across different sites. Compare that to just 8% of CD8 plus T cell clones. So what does this actually mean for us? It means that, unlike T cells, which tend to just hunker down and multiply locally, these innate myeloid cells are highly mobile commuters. They're constantly moving out of the blood supply line and infiltrating the tissues.
That brings us to Chapter 4, the DC3 lineage connection.
Here is where we introduce a very specific person of interest into our detective story, the DC3 cell. DC3s are a unique type of dendritic cell that are highly enriched inside tumors, and they play a massive role in whether the immune system ultimately succeeds or fails. Now, scientists have debated for a long time about where these tumor-enriched cells actually come from. By mapping their UMAP clustering, which is basically where shared features group together visually, and combining that with Mitotrack's family trees, the researchers linked these tumor-resident DC3 cells directly back to everyday circulating monocytes in the peripheral blood. So this builds a complete, undeniable origin story for the DC3. The data shows that DC3s aren't actually born inside the tumor, nor are they related to other classic dendritic cells. Instead, they're directly supplied by the blood. They actually retain the epigenetic memory of being a monocyte, sharing the exact same transcription factor motifs like AP1 and MAF. They are, without a doubt, just everyday circulating monocytes that have stepped off the supply line and then transformed into DC3s right upon arriving at the tumor. And here is where things get really crazy. Chapter 5, the plot twist, fate bias. Literally everything we've covered so far leads to a discovery that completely upends a major scientific paradigm. The researchers concluded, and I quote, intratumoral myeloid differentiation fate may be peripherally programmed before TME infiltration. Guys, this is massive. The old medical assumption was always that a naive immune cell enters a tumor, and then the local tumor environment dictates its behavior. It tells it whether to be a helpful cell or a harmful cell. Well, this study proves that assumption is just flat out wrong. It turns out the monocytes in your bloodstream already have their orders before they ever even touch the tumor. Researchers found two completely distinct family lines circulating in the blood. The first are DC-biased monocytes. These are pre-programmed with pro-inflammatory genetic signatures like IRF and STAT2. They're primed and ready to become helpful tumor-fighting dendritic cells. But the second group are macrophage-biased monocytes. These are pre-programmed with immunosuppressive signatures like MAF and NRF2. They are destined to become harmful macrophages that actually protect the tumor. Their fate is completely sealed in the blood, not on the battlefield. Now, what's really interesting about this graphic is how perfectly it visualizes all of this. Over in the peripheral blood on the left, we can clearly see these pre-biased monocytes. Before they even infiltrate the tumor microenvironment on the right, they are already carrying the specific transcription factors that dictate exactly what they're going to become. The supply line itself is receiving systemic programming.
Which brings us to our final chapter, redefining cancer immunity.
You know, this isn't just some interesting quirk of biology. It has absolute paradigm-shifting implications for how we're going to treat patients in the future. So the crucial point to take away is this. The tumor myeloid compartment is peripherally programmed, not just locally trained. Tumors aren't just fighting a localized battle. They might actually be sending out systemic signals into the entire body to essentially brainwash the immune supply line before the troops even reach the battlefield. They're ensuring that the reinforcements arriving from the blood are already programmed to help the cancer survive. Which leaves us with one incredibly provocative question. If tumor immunity is actually being shaped out in the blood before the cells ever even enter the tumor, can we intercept that process? Can future immunotherapies actually program the supply line in the bloodstream to guarantee we win the war? The possibilities here are absolutely staggering, and it opens up an entirely new frontier in cancer research. Thank you so much for joining me on this explainer. I really hope this sparked your curiosity to learn even more about the incredible world of our immune system.